Hello, everyone, and welcome to our Cell and Gene Live, redefining cell and gene therapy with exosomes and EVs. Thanks for taking the time out of your day to join us for what promises to be an insightful and engaging discussion. As Heather said, I'm Erin Harris, Chief Editor of Cell and Gene, and I'm excited to be joined by Dr. Gloria Matthews, Chief Medical Officer at Aegle Therapeutics. Dr. Matthews is an executive surgeon scientist with nearly 3 decades of expertise in biomedical innovation and product pipeline development. And we're looking forward to her perspectives on the science, challenges, and clinical promise of extracellular vesicle-based therapies. Unfortunately, Dr. Linda Marbon is unable to join us as originally planned. Some exciting things are happening at Capricor, and so we wish them all the very best. As always, we want this to be as interactive as possible, so please don't hesitate to submit your questions for Gloria using the Q&A tool. We'll get to as many of them as we can throughout and at the end of our discussion. And as always, we have poll questions that we would love for you to provide your responses to so that we can kind of talk about them as we go through as well. All right, so let's get started. So first of all, Gloria, welcome. Thanks for your time today.
And you, Erin, it's my pleasure.
Okay, so Aegle Therapeutics stands out as the first company to receive FDA clearance for an EV-based therapy with a lead program targeting severe dermatological conditions. So among all the other things you're working on, can you briefly introduce Aegle's unique approach and to leveraging exosomes and EVs in therapeutic development and the specific indications you're targeting, even and how that impacts the cell and gene therapy space?
Sure, I'll be happy to. So at Aegle, we are working with bone marrow-derived mesenchymal stem cell EVs, extracellular vesicles, which includes all of the vesicles that bleb from the mesenchymal stem cells. So it includes things like exosomes and apoptotic bodies, microvesicles, et cetera. So it isn't just strictly exosomes. We think there might be some advantage to that in terms of just having a greater milieu, and I'll talk more about the milieu as we go through, of factors that come from these stem cells that can help drive regenerative healing. So that's our main focus, is regenerative healing at this point. And we also are very interested in the way that EVs can, I'll call them EVs, extracellular vesicles, the way that they can modulate the immunoinflammatory response in certain diseases. So one of our first indications that we went after in the clinic was in conjunction actually with the Department of Defense is burns, deep second-degree burns, trying to help to prevent the conversion of deep second-degree burns to third-degree burns, and also to help the healing, not just healing, because these wounds do heal. Deep second-degree burns eventually heal. It takes a long time, and they don't often heal with a very cosmetic or functional result. So there's frequent, hair, there's deep scarring, pigmentation, the skin is rougher or tighter. There can be constrictions over joints, et cetera. So really is not just a matter of whether something looks nice or not. It's actually functionally can be very impairing. And so we ended up treating our first patient recently and got extremely good results after a week. So they were allowed to get multiple doses over time each week, and that patient only ended up getting one because his wound completely closed. He had a similar wound. This was from a gasoline fire explosion in a boat. Similar wound on the other foot, and it really didn't heal anywhere near as nicely as the one that was treated. Now, we didn't have a formal control. So in our next study that we are doing now, epidermolysis bullosa, which is a severe blistering disease from birth, that's a rare genetic disease. It's a deficiency of collagen 7, but it's also a dysregulation of the immune system. And so there's not only just missing Col7, which can be replaced, and there is actually, Crystal Biotech has very recently gotten approved a product that is a Col7 gene therapy replacement for wounds associated with epidermolysis bullosa, and they've had some considerable success with that. That does not, a Col7 alone therapy is very helpful, but it doesn't address some of the other issues that are going on locally and also systemically in terms of the immune dysregulation and responses to not having collagen 7. So we uniquely think that something like EVs can do that and do that better than just a COL7 alone therapy, potentially. So we have looked at the things that are in our EVs, and we see that it's very much reflective, and they're not all exactly the same from lot to lot. So these come from allogeneic donors of these stem cells. So they're not all exactly the same, but when we did a proteomic analysis of these little vesicles, we found that the categories were extremely conserved. So you'd have different members within the categories, but yet the number and type and magnitude of the response within each category of protein type was amazingly conserved from donor to donor. And what our scientific co-founder had found early on, who's a dermatologist and dermatopathologist at the University of Miami, is that many in his animal models and even in some clinical work that he did, that many of the, or all of the effects that you see with mesenchymal stem cells, which we know have been pretty positive but hard to demonstrate in regulated clinical trials, those are actually can be attributed to the EVs alone that bled from the cells. And so he had started some early clinical work with just isolating the extracellular vesicles and finding best ways to do that, and had found some promising data. And we modeled a lot of what we've done from that early data. So I think that does help to kind of set us apart in this approach when we're looking for regenerative healing. I think that's the main thing. We're not just looking to close the wounds or fix the issue. We're actually trying to make it so that it's more like it was when it was normal.